Over the past few summers, I had a lot of time to kill. And sometimes it was done pretty well. I would study for classes in the next year. I would do Coursera courses online. I would code. I would read. And sometimes it was just involved binge watching a bunch of shows. And one in particular that stood out to me was this one really cute anime called Cells at Work. TLDR, Cells at Work was about a bunch of cells in the body, but specifically, it was about blood cells and immune cells. We're actually going to be focusing on the immune cells today. And TLDR, your immune system is basically the army in your body that protects you from germs and whatever harmful things that may occur within. So think cancer, viruses, bacterial infections, prions, you name it. And yeah, that it prevents stuff like that from happening within, which is kind of creepy. So they're real fighters, and you should really appreciate them. And there are a lot of cells that are actually involved in this process, but um, sometimes they can be like very widespread and can kill a bunch of different cells, like neutrophils. And sometimes they can be really specialized in killing these infectious cells. And we call those T cells. So there are two different kinds of T cells, and that is shown in this picture that I took from the anime. So the guy on the left is a helper T cell, and he's basically like the Houston, Texas of that. He tells the other cells, okay, this is what bad guy I should probably end up killing. The other kind of feral and psycho looking guy is a killer T cell or a cytotoxic T cell. And this is the T cell that we kind of want to target. So the thing is, they can't always properly tell apart that, okay, this is a good guy and this is a bad guy. Sometimes they end up confused, just like you or me. So we have to make sure that they can actually tell them apart and kill them, or else um, crazy stuff like this will happen, and we kind of don't want that to happen. So what we need is the perfect killer T cell, and that's where engineering and gene editing come into play. We have to engineer these T cells so that they'll be able to perfectly recognize and then go and kill these bad cells. In the specific case of cancer, cancer cells have this specific protein on their surface called an antigen. And what that scent analogy that I described earlier, um, if the CAR T cell will be, if the T cell will be able to bind in a lock and key formation, because it's very much like a lock and key. It only works together. They can't go with anyone else. If they can do that, then they can wrap around and kill them. So what we're trying to do here is engineer a chimeric antigen receptor onto a T cell. And these receptors essentially can bind to any specific antigen and can tell it apart. And then they'll be able to kill it. It's the ultimate solution, and it's coming from within your body. And note this, loads of companies are taking this approach. However, I'm going to be focusing on a specific one from CRISPR-TX. So, CRISPR has been a gene editing technology that has been used for a while, ever since Jennifer Doudna and Emmanuel Charpentier had discovered it. And they essentially described the usage of it in many practical applications as just a slice. It can cut genes at different locations, and it can also add in different genes. So the way that we're going to use it in the case of CAR T cell insertion is mainly to disrupt and then insert the gene. So how does this work? That cut up um, part of the gene which is because of the CRISPR technology. And then with um, additional usage of CRISPR, we have insertion. So, implementing these principles, the company uses these three in their three-step framework. First, they use deletion in order to remove the really natural T cell receptor, and it's not very efficient. So if they're going to be replacing it, then why have it? Second, they're going to insert the chimeric antigen receptor coding gene into that specific location. And that will mean that instead of the TCR original protein being expressed, this specific chimeric antigen receptor will be expressed. Finally, they will delete the major histocompatibility complex. And basically what this does is that because it'll recognize that these T cells are coming from outside, they will facilitate an immune response, which will go against and make the tumor a lot more harmful. So removing this will make sure that this effect won't happen. CRISPR Therapeutics has stated that they are following an allogeneic pathway rather than an autologous pathway. So with an allogeneic pathway, they harvest the T cells from a healthy patient and then they bring it to the manufacturing factory so that the scientists can engineer it with the three-step process described before to express for CAR T cells. And then it'll be brought to the patient in need. However, with an autologous pathway, it'll take the patient's own cells and then re-engineer it and then bring it back to them. However, it'll take 2 weeks after harvest for this to happen, whereas it could be pre-made and ready beforehand. However, they will recognize that the cells will recognize that this is a different cell from the one in their body. So that's why we had to get rid of the major histocompatibility complex. Right now, there are two different therapeutic programs that are in usage for CRISPR therapeutics in terms of immuno-oncology, with CTX 110 used for B-cell malignancies. So this is whenever the cancers have spread to the lymph nodes, and they also use CTX 120 for multiple myeloma, which is the malignancy of plasma cells. So this is very much involved with blood and immune cells. However, they are actually transitioning to solid tumors with renal cell carcinoma. The antigen being targeted in CTX 110 is CD19, whereas it is BCMA for multiple myeloma. With this video, I went into a little bit of a deep dive on how immuno-oncology and the whole system behind CAR T cells work. And I also added some insight into how CRISPR Therapeutics is using gene editing in order to implement this policy. I do think that this is going to be the future of anti anti-cancer drugs and therapy, because this is using your own system for you rather than it somehow malfunctioning and going against you, as has been the case for so long. I really hope you gained some value from this video, and if not, please drop some feedback and give me a like and subscribe. Thank you!